Hello everyone, welcome back to the channel. Today we are discussing about quantitative microscopy of crude drugs. Today we are dividing into microscopic techniques used in the evaluation. First is quantitative microscopy with Lycopodium spore method. It's a key tool for precise particle analysis. Next we have leaf constant, essential for botanical studies. Finally camera lucida, helping us examine fine details at a microscopic level. Lycopodium spore method is used when chemical and other evaluation methods fail to determine drugs quality. It's commonly applied to powder drugs like clove, ginger, cardamom, nutmeg, etc. These methods help estimate the percentage of foreign organic matter in powder drugs. Lycopodium spores obtained from Lycopodium clavatum in 1 mg of Lycopodium contains an average of 99,000 of spores. This technique ensures precise drug evaluation. For the Lycopodium spore method to work effectively, certain conditions must be met. First, particles should have a definite shape and be easily countable like pollen grains or starch grains. Secondly, if a single layer of tissues or cell is present, the actual area covered by spores can be calculated precisely. Lastly, the parts being measured should have uniform length and thickness, like trichomes, ensuring accurate evaluation. These conditions help make the Lycopodium spore method a reliable tool in microscopy. Let's quickly learn how to evaluate drugs using the Lycopodium spore method. First of all, what are conditions for evaluation? For this method, particles should have a definite shape like pollen grains or starch grains. If there is a single layer of cells or tissues, we can accurately calculate the spore covered area. The parts being analyzed must also have uniform length and thickness such as trichomes. First, determine the moisture content of the powder sample. Then weigh and mix the required quantity with Lycopodium spores. Suspend the mixture in a viscous liquid. Then mount a drop on a slide and it under a microscope. Count the number of Lycopodium spore and characteristic particles in 25 yields. Repeat with a second similar suspense. Then what is the formula? Percentage of purity of drug equals to N into W into 94,000 by S into M into P into 100. So what is N? N is number of sample particles in around 25 yields. W denotes weight of Lycopodium taken in milligram. S represents number of Lycopodium present in spores. M represents sample weight. P represents number of characteristic particles present in per milligram of drug sample. So N, W, S, M, P all are sample specific values. This method ensures precise drug evaluation. Let's quickly break down how to determine leaf constant in botany. This includes stomatal number, stomatal index, vein islet, vein termination number and palisade ratio. The stomatal number is simply the average number of stomata per square millimeter of the leaf epidermis. The stomatal index tells us the percentage of the stomata compared to the total number of epidermal cells. The formula is stomatal index equals to S by E plus S into 100, where S is the number of stomata per unit area and E is the number of epidermal cells in the same unit area. Did you know that tiny veins in leaves help scientists identify plaques? Let's talk about vein islet and vein termination number. In 1929, Levin introduced the concept of the vein islet number, which is the number of vein islet per square millimeter between the midrib and margin of the leaf. Later in 1951, Melville defined the Venlet termination number, which counts the Venlet terminations in the same area. These values help in plant classification and identification. Did you know that tiny leaf cells help identify plants? Let's talk about palisade ratio. In 1925, Jorning and Weiss introduced the palisade ratio, which is the average number of palisade cells beneath each epidermal cells. For example, the Venn islet number of Alexandria senna ranges from 25 to 29.5. This helps in plant identification and classification. Camera lucida is an optical instrument used with a compound microscope to trace magnified images. It has three main components. First is the attachment ring which connects to the eyepiece, and then the microscopic image and a mirror set at a 45-degree angle and is reflected from the prism to the mirror and then onto the drawing surface, allowing precise tracing. These tools helps in scientific illustration and microscopic studies. It's all about quantitative microscopy of fruit drugs. For more details, please check out the link in the description below. Don't forget to like, share and subscribe for more study tips.